First off, we're going to discuss why computational techniques are very useful for ion channels and transporter protein in general. Well, firstly, if you think about structures and how do we understand ion channels and transporter structurally, right? We need to obtain the structure, right? So we need to go and obtain the crystal or cryo-EM structure. Back in the very old days, obtaining crystal structure, and still now actually, are very, very difficult and time-consuming and expensive process. Well, it's cheaper now, but it's still quite, expensive, especially for crystallography, where you need a lot of protein yield. So you have a yield problem, which is now solved by cryo-EM, but there are still problems with those. So only until recently that we have a lot of structures available. So what have we been doing back in the old days? Well, what we have done is, based on the existing structure, computational tools allow us to model the structures based on existing structure. So using the crystal structures, it allows us to model the structure based on the existing one, right? And that's the idea, especially with several proteins. Now we know that it has multiple conformation. So for example, ion channels can exist between a closed and an open state. We might have a structure of a closed state channel, but the open state is very dynamic and it would require great effort to get the structure. What we can do before or while waiting to get the open state structure, what we could do is try our best effort based on existing structure to model an open state structure and use that to hypothesize or guide experimental study to work out the function as it transits from closed to open. What is required in the gating process of these structures. When we obtain these crystal or cryo-EM structures, sometimes we've got structures with the blob of lipid, but we're not very sure whether that is lipid or which lipid is bound, or is it just detergent sticking there. What we can do is we can take these protein which are in the crystal, a static protein in the crystal, and then do something called molecular dynamic simulation, put it back into their membrane environment and see how they behave in the membrane bilayer in the simulation box. The detail of how we've got to put them in a simulation box, we're going to discuss later, but we can use molecular dynamic simulation as a tool to observe the behavior of membrane protein of our interest in the box. It will also allow us to fix the structure. So, we get the structure, we do MD, put them in a box, but if our structure beforehand has been mutated to be thermostable or being a bit chimera, we can work on this and fix our model using the sequence of a known structure or sequence of our protein. So actually, before we put our protein in the box, we kind of need to fix our structure and put them in before we put them in the box. So this can be our first PDB structure, structure one, and then what we have is one hash, which is the fixed one. Also, what we are also working on now in a molecular simulation is something called patch clamping in our computational electrophysiology, which means that normally when we do a wholesale patch, right? So we have a cell, we have a pipette at the tip of a cell, and we try to record a current, and we excite the patch. So we got this little piece of membrane here with an ion channel. What we can do now as well in a molecular simulation is having a box with two layers of bilayer. So you can have two environment, A and B. B is connected from here. We will discuss how why B is connected later. And we can have an ion channels here where we can vary the voltage as a concentration of ion difference from A to B and use that to record permeation of ion. And this is what we call computational electrophysiology as a technique. So, MD can provide several ventures of looking at the dynamic of a protein and the molecular behavior of an ion channels, almost like a microscope to look at a single molecule microscope to look at an ion channels or molecules behavior in the membrane. And it is not just that. The other problem that we can use MD to look at is lipid behavior. So, sometimes you get a structure like this and you might have a blob of lipid, right? And we would like to identify what lipid it is that is bound to the structure. So we can run simulation and see the type of lipid that might come and adhere to the binding site here of our interest and use that as a way to identify the identity of the lipid, their binding site, and also their binding affinity. The details of all of that we'll be discussing later on in the tutorial.